Okay, next we want to get into the eukaryotic organelles in a little more detail. As I mentioned, this is one of the two defining features of a cell, the internal organelles. And as we already mentioned, each is its own kind of independent functioning part of the cell, kind of like how organs have their own independent functioning within our own body as a whole. Now, this here is a figure of a eukaryotic cell right out of your textbook. This figure is meant to show all of the various features of a eukaryotic cell without saying that this is exactly what they look like. So this is not what all eukaryotic cells look like. In fact, probably most eukaryotic cells don't look like this. So don't take it as this is what they all look like, but rather this is a figure that we can look at to kind of get an idea of what they look like, and it's more or less to point out some of the different features that we'll talk about here as we go. If you remember from chapter 4, we had a similar feature or similar figure of a prokaryotic cell shown down here that was kind of the same thing, meant to give a generalized overview. The difference is that this one doesn't have any descriptions of the features like we saw in chapter 4, but similar overview figure. Now, the nucleus is one of the two defining features of a eukaryotic cell, but it's also actually considered an organelle. I know, go figure. The difference is that it's the most significant and most fundamentally characterizing organelle of the cell. So it's actually kind of specified in that definition as its own kind of thing, even though the truth is it's technically part of the organelle group, like other organelles, like the mitochondria, like the Golgi apparatus that we talked about earlier. So it's still an organelle, but it's kind of like the king organelle. And so they separated that out as a separate feature. And it's what houses the cell's genetic material, as I already mentioned. Here you can see some figures. These are both animations, and this is a real figure that's got some artificial coloring to it. But here you see some cells where you can actually see the membrane. This loose kind of boundary here is the membrane. And then they've basically just highlighted the internal nucleus to show how that would kind of look in the cell. You can also see some of them are different size, some of them are bigger, some of them are smaller, so they're not all the exact same size, but they do take up a fairly good chunk of a eukaryotic cell, and they're kind of one of the more prominent features of a eukaryotic cell. Here's another animation or 3D illustration of it. And this down here, I'm not sure if these are nerve cells. They kind of look like nerve cells. I'm not sure about that, but you can see these are real cells. And here you can see the nucleus taking up a big part of that. These are artificially colored, so the nucleus isn't normally blue like that. It's been stained to show, it's actually a fluorescent microscope image that makes it kind of stand out from the rest of the cell. So here it states that the primary purpose is to protect the DNA and also help regulate what goes in and out. Kind of like how our brain and our spinal cord kind of help protect the skull and the spine kind of help protect the brain and control what goes in and out. The nucleus is itself kind of one thing, but it's made up of three distinct components, at least three, and we're just going to focus on the three major components. One is actually called the envelope, the nuclear envelope, and that is what actually separates the nucleus from the rest of the cells. This is that kind of separate compartment, the separate covering that separates it, kind of like the skull of your brain. Nuclear pores are what allow things to come in and out, almost kind of like how the spine allows information to come in and out through nerve cells. And then the nucleolus is almost kind of like the major stuff of the nucleus, almost kind of like the brain tissue to a certain extent. It is actually what really makes up a lot of the core functions of the nucleus, and it's actually where ribosome synthesis takes place. So we said ribosomes are a part of what makes protein in the cell. Well, they themselves are created within the nucleus, so you kind of have a process of generating those within the key part of the cell. Not too much detail there, just recognize those three parts and the basic idea of what each does. Okay, so next I want to talk about what's inside the nucleus in terms of the DNA. So as we mentioned earlier, DNA is found in the nucleus. It's kind of the key idea of the nucleus is to protect and regulate the DNA. So even though that wasn't mentioned on the last part, I want to talk about that now. And I also want to talk about how it all fits in there. So the DNA is kind of its own separate topic, and we'll talk about that when it comes to Chapter 9 later in the semester, what it does, how it functions, and all of that. So for now, we're kind of talking about more of the structural parts of it. So that's kind of why I'm focusing on the idea of DNA compacting. So the idea is that we have all this DNA in our cells, and in a eukaryote, there is an especially large amount of DNA. So take a human being, for example. Humans have 46 chromosomes, and of those 46 chromosomes, if we were to stretch out the DNA of each chromosome end to end, we would find a total of about 6.5 feet of DNA per cell. Now, in your body, you have approximately probably 40 plus trillion cells. Those estimates vary. It all depends on how big you are. But roughly 40 trillion cells. And in every single one of them, minus your red blood cells, so that I guess maybe a slight exception there, but red blood cells don't have DNA within them, ironically. But despite, with the exception of those, every cell has approximately 6.5 feet of DNA in it. And they're so small that you could fit several thousand on the head of a pin. So, kind of mind-boggling to think about that. So, the only way that they get that much DNA within a cell is to tightly wrap it and package it and condense it into these really, really, really tight and highly organized bundles. And that's what we're talking about here. And this doesn't really happen in a prokaryotic cell. Prokaryotes have significantly less DNA and have a much less refined way of wrapping it. They kind of do condense it, but nowhere near on this level. So the way this works is we have specialized proteins called histones. Histones shown here are these little bead-like proteins. And what ends up happening is the DNA, which starts in a thread-like material, actually wraps around those histones, and then those histones kind of group together, as you see here, and then they wrap in tight and coil even further. And then ultimately these wrap into the structures that we know as chromosomes. So what you ultimately see is a wrapping and a folding and a condensing process that allows the cell to take 6.5 feet of DNA with, as an example in humans, and then condense that down until it all fits in a cell so small we can't see it with the naked eye. So that's how the DNA actually fits in there. So that's the last thing that kind of goes along with the structure. So within the nucleolus here, around the nucleolus, so between the membrane and the nucleolus, we would see all that DNA compacted in there. And they don't show it here because it would basically just fill every square inch of that nucleus. Okay, next part we're going to talk about is called the endoplasmic reticulum, abbreviated as the ER. Endoplasmic reticulum is what branches off from the nuclear envelope. And I mentioned that earlier in the drawing I made. It's a series of flattened membrane that is continuous with the nuclear envelope. So here you can see it in this figure. Here's the nuclear envelope. What ends up actually happening is it actually folds outward and starts to create a whole new structure that we call the endoplasmic reticulum. So it's almost like this extension of the envelope of the nucleus. And within it, what it ends up doing is forms a series of tunnels that you can see here that kind of connect and branch out from the nucleus itself. Now, as it branches outward, it actually differentiates into two distinct types. One is called rough endoplasmic reticulum. The other is called smooth endoplasmic reticulum. And so here you can see in this figure, on one side over here, we have rough endoplasmic reticulum, and over here we have smooth endoplasmic reticulum. The difference is that rough endoplasmic reticulum has the ribosomes I mentioned earlier. Ribosomes are what give it a quote-unquote rough appearance. It's just a total arbitrary way of naming it. Somebody decided that's what they wanted to call it. It does make sense, but that's what they went with. Now, ribosomes, as we know, function in protein synthesis. And so what we see is that with those embedded in this part of the membrane, we end up with a structure that facilitates protein synthesis. So in other words, rough endoplasmic reticulum basically is a structure that organizes the ribosomes and collectively helps the cell produce proteins. So that's really what its major function is. It kind of supports the ribosomes, which then make the proteins. Now, in addition, it also helps transport those proteins, and it also helps kind of polish them to a certain extent. So there's kind of some fine touches that go onto them, and then it also helps kind of send those out to other parts of the cell, as we'll see here in a minute. So, but for now, just think rough endoplasmic reticulum, RER, think ribosomes, think protein synthesis. Now, other parts of the endoplasmic reticulum are referred to as smooth, SER, and they're considered smooth because they lack the ribosomes. So there's really a distinct difference in either whether you have ribosomes rough or don't have them smooth. Now, the functions of the smooth endoplasmic reticulum are a little more complicated and not something that I want to go into too much detail here with, because you kind of open up a can of worms if you try and elaborate too much. You could really spend a lot of time focusing on that, because the functions are fairly diverse and there's a lot of variation depending on what type of cell you're talking about. So, for example, your liver cells in humans, the SER is very different in terms of the exact functioning than it might be in, say, your brain cells or in the reproductive cells. There's very different functions. For example, in the liver, SER has a more specific function of detoxification, whereas in, say, the reproductive cells, they function largely to make things like estrogen and testosterone. So, again, don't want to go into too much there, but they do a lot of processing, and this involves largely lipids and things like that, which explains some of the functions there. So for now, just recognize that they're smooth due to a lack of ribosomes, and that's really all the more I want to get into there. Just to clarify, though, from the study guide, when it says what are the functions of each, for the SER, I'd like you just to know that they function in nutrient processing and synthesis, in storage of non-protein molecules such as lipids. Okay, so that line right there kind of tells you what's the function of the SER. Be a pretty easy multiple choice type question. I won't go into too much more detail than that. Here's a picture showing the endoplasmic reticulum, a different picture. Here, this is out of our textbook, and this here is showing a close-up of the ribosomes embedded within there, kind of how they would be functioning. And then over here, you see an actual, what's called a transmission electron microscope image of the endoplasmic reticulum within a real cell. So here's the nucleus, and here's the different compartments of the endoplasmic reticulum. Okay, the next organelle is called the Golgi Apparatus. And the Golgi Apparatus is one that is often referred to as the quote-unquote UPS or post office, or maybe even the FedEx of the cell. Now, one question I like to ask is why is it called this? And in fact, that's right off the study guide. Why is the Golgi Apparatus often referred to it this way? The reason is because the Golgi Apparatus largely functions to receive substances like proteins and lipids created from the endoplasmic reticulum. So it receives those products and then redirects them as to where they're supposed to go. So it says here, it's the site in which the cell, when proteins are modified and then sent to their final destination. Protein synthesis usually begins in the RER, the rough endoplasmic reticulum, and completes at the Golgi apparatus. So in the Golgi apparatus, what's happening is that proteins are being sent from the endoplasmic reticulum once they're produced into the Golgi apparatus. Now let me find a figure here of that. Here's a figure. So over here we see the endoplasmic reticulum. What ends up happening, I'll talk about how this happens here in just a second. What ends up happening is the products, like the proteins, are actually packaged up in these little things called transitional vesicles, and they're then sent across the cell and then are received by the Golgi apparatus, which is what you're looking at here. This yellow gold structure is the Golgi apparatus. So these proteins then are fused into the Golgi apparatus, and then they're literally modified and then repackaged as they come out the other side. So over here, they're actually transitioning out of the Golgi apparatus. And inside here, they're not shown here, but inside here, you would now have kind of modified and then repackaged products that are ready to be sent to wherever they're supposed to go within the cell. And on this next slide here, you can see a better overall figure of how that works. So here is the cell's nucleus. Here are the ribosomes within the endoplasmic reticulum. Here you're seeing some of these vesicles budding out. They would actually travel across the cell and then fuse with the Golgi apparatus. The products would migrate through the Golgi apparatus and be modified, and then come out the other side and be repackaged, and then be ready to be sent somewhere within the cell. So what ends up happening then is that some of these vesicles will then migrate to the cell membrane, and then the products within here would actually be dumped outside the cell. So the Golgi apparatus, for example, might be involved in taking a protein that's made inside the cell and then secreting that outside the cell. An example of where that would happen in your body would be your neurotransmitters. Things like dopamine, serotonin, probably familiar with those. These are neurotransmitters that help our brain communicate. Well, those are proteins, and the proteins are made inside the cell, but they function outside the cell. So the way that they get outside the cell is through the Golgi apparatus. They're first made here, transported, and then sent to where it is they want to go. In this case, they want to be secreted outside the cell, and then they would go on to have an effect kind of in between two cells. You may be familiar with kind of how your nerves work, axons and dendrites and things like that. So these would kind of go from one nerve cell to another. In between there would be outside of the cell, and they would help communicate. Now, also not shown in this figure, these vesicles can also be sent back inside the cell, and many of them will function internally. So the Golgi apparatus will kind of determine whether they go outside the cell for secretion or whether they're brought back in the cell for an internal processing. Okay, and that's not all that detail is not shown here, but that's kind of the idea. So this is what's referred to as nature's assembly line. The idea is that proteins, or rather the information in the cell, begins in the nucleus, extends into the endoplasmic reticulum, branches to the Golgi apparatus, and then ultimately is directed somewhere within the cell, either internally or externally. Let's just go back here real quick. That's why it's referred to as the UPS. Kind of like the UPS, you take them a package, they put a label on it, and that label determines where it goes. That's largely how the Golgi apparatus works. The proteins in this case are sent. The Golgi apparatus literally puts a biological label on them, and then that label is what directs them to wherever they're supposed to go. The only difference with the Golgi apparatus is that the Golgi apparatus actually modifies the package as it's being directed. Hopefully the UPS isn't modifying your package too much. So that's one difference. But in a sense that it receives, labels, and then sends, the Golgi apparatus can be thought of as the UPS. So that's a question I may just ask you in short answer. I don't know, but you might want to be ready to explain that in a couple sentences. Now, one more thing, this is also true of the SCR. Just to go back here real quick, SCR, as we said, produces things like lipids. So some of the lipids are processed in the same way that proteins are. So these vesicles here could be branching off the smooth endoplasmic reticulum, in which case they would carry lipids as opposed to proteins. So really the Golgi apparatus could receive either proteins or lipids mostly, and then modify and redistribute those. Okay, hopefully that makes sense. If not, you might want to go back and look at that again. Always feel free to ask me questions. There are some really great videos on YouTube as well, and I try not to show those in here as much because I know it takes a lot more time. Now, one way that the cell actually sends things is through what are called cellular vesicles. Over here, they were referred to as transitional vesicles. It's kind of two ways of saying the same thing. I know that gets confusing. I apologize. The term vesicle is what's key here. The way that works is that you have membrane that is made of those phospholipids. So if we just go back here to refresh your memory, we showed phospholipids all the way back here. These phospholipids are very flexible and kind of squishy. It's almost like Play-Doh. Whereas if you were to squeeze this phospholipid, you can almost pinch part of that off and separate it. You could not almost, you literally, if you had microscopic fingers, you could literally grab a hold of that phospholipid and just take a piece of it off. Just rip it right off. And like Play-Doh, the rest of it would kind of fill in around it, and the cell would kind of reshape itself to compensate for that missing piece. It would literally kind of fill in the gap automatically. And then that piece could literally be pulled off. It would actually reform a new vesicle like this. It would actually go back into this kind of form where you've got the membrane heads on the inside and the outside. And you would literally create a separate little vesicle that was pinched off from the rest of the cell. So that's what's happening when we look at a cellular vesicle. What's happening here, this is a part of the membrane. Just go back here. Like this is what you're seeing, like right here. So here is this is a part of a phospholipid membrane. So the phospholipid membrane is both on the inside and the outside of the cell. I didn't say that just to clarify. It's a slightly different construction, but similar type of material. So you can literally pinch part of that off, which is what's happening here. And then as it rips off entirely, here's one that's in transition. the membrane would just kind of reform and reseal itself. Within here, you would have some of the products being produced by the rough endoplasmic reticulum. So now those literally float across the cell and then refuse with the Golgi apparatus over here. And that's kind of what you're looking at here. So here's one budding off. You can see these little green specks have the products within them, like the protein. They physically move across the cell and then fuse with another component over here. And here's another figure from a side figure, a cross-sectional illustration showing the same thing. Products gather near the edge, it pinches out, floats across, refuses, and the products get merged into this secondary location over here. So this is how products get from the endoplasmic reticulum to the Golgi apparatus, by literally budding out, floating across, fusing, and then dumping those products into the Golgi apparatus. If you take it for what it is, it's relatively straightforward. If you think about it on a more in-depth level, this is pretty complicated stuff, and it's hard to explain exactly how that works, and we don't have the time to do all that. So, there's videos online that will give you a more in-depth overview of how this works. But for this class, you just kind of take it for what it is. These are basically little transitional membranes that carry products from the endoplasmic reticulum to the Golgi apparatus. And then, just to clarify, these are the same kind of things that would bud out from the Golgi apparatus and secrete things outside the cell or inside the cell. It's a little flexible membrane that can bud off and fuse on the other side of things. So the key thing here is that this is what is used to transport things throughout the cell. There's an example that I want you to be familiar with of a type of vesicle that goes on to have a more specific function. So there's more detail here than what we're going to cover, but I just want to add one little thing to it. So there's actually a certain type of vesicle that is modified in a way that gives it a special purpose, and these are called lysosomes. Lysosomes are a type of vesicle that contain digestive enzymes. So what they end up doing is being infused with a type of enzyme that will literally break down food-like substances into smaller pieces. Just like our stomach digests food, these are basically like an internal stomach of the cell. In fact, this is actually how your body digests things inside of its cells, which happens every day. So you can almost think of it like a little micro stomach for the cell. And what you're looking at here is a figure of how that works. You see the Golgi apparatus here, and you see these little lysosomes budding off of those, and they have these digestive enzymes inside them. As food particles are brought inside the cell, the lysosomes fuse with the food particle vesicles, and then those lysosomes go on to break down that food-like material. And then the energy from that can be redistributed throughout the cell, and then the waste can be eliminated through a process called exocytosis, where the vesicle buds outward and dumps that contents outside the cell. Here's another figure showing the same thing. Here's the food being brought in its own little vesicle. The lysosome merges or fuses with that, and inside there the food is digested by those enzymes. So that's just one example of a handful that we could talk about of a cellular vesicle that has a function of digestion. Also, we'll talk about this later in chapter 14. We'll talk about how this is modified in our white blood cells to actually be a component of digesting invading microorganisms. So we talked a little bit about phagocytes in chapter 4. Phagocytes are your white blood cells that digest and kill microorganisms. Well, one way that they actually kill the microorganisms is by using lysosomes. So in some cells, this is a food that's digested. In other cells, it's actually an invading microorganisms like bacteria and fungi and algae and things like that. Or not algae, but yeast is what I meant to say. So I know this is confusing. I know it's a lot to take in, but this is why I have the study guide. Stick to what it is. The questions are asking. So what is the function of a lysosome? It is to digest, and it is basically a special type of cellular vesicle. Okay. Here's a quick video of the lysosomes.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles, The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
Okay, so they're pointing out another feature I didn't mention, which is how these can actually be used for internal housekeeping, so to speak. Break down worn out organelles and allow the cell to generate new ones to replace them.